Welcome to the Pharmcogenomics program, Principles and Concepts of Pharmacogenomics. My name is Dr. Grace Kuo, and I will be presenting the information in these slides along with my colleagues, Dr. Cindy Gustafson-Brown and Dr. Joe Ma. The presentation is divided into several sections outlined on this slide. The first 5 sections explain foundational concepts in pharmacogenomics. Next will be ethical, legal, social, and economic issues, followed by roles and resources for healthcare professionals. Upon completion of this program, we expect participants to be able to describe and define basic pharmacogenomic nomenclature and principles, describe polymorphism types and their impact on pharmacokinetics and pharmacodynamics, understand the ethical, legal, and social issues as well as economic issues related to pharmacogenomic testing, and identify resources for obtaining current and updated pharmacogenomic information. First, it will be helpful for us to review definitions of pharmacogenetics and pharmacogenomics. Pharmacogenetics is defined as the study of genetic causes of individual variations in drug response. Pharmacogenomics encompasses a larger scope. It more broadly involves genome-wide analysis of the genetic determinants of drug efficacy and toxicity. These definitions have been proposed by the different regulatory agencies and professional organizations, and the terms are usually used interchangeably. For the purposes of this presentation, we will use the term pharmacogenomics. We'll begin with translating pharmacogenomics into practice. Currently, there are persistent problems with drug therapy. Drugs used for many common diseases such as Alzheimer's, depression, hypertension all have drug response rates which are only between 30 to 60%. Adverse drug reactions are quite common. They can increase morbidity and mortality as well as the cost of care. Variations in a gene may impact either the pharmacokinetics or dynamics of a drug, which in turn affects the clinical outcome. Recall what pharmacokinetics and pharmacodynamics mean. Pharmacokinetics is the process by which a drug is absorbed, distributed, metabolized, and eliminated by the body. In essence, what the body does to the drug. Whereas pharmacodynamics is the action or effect of a drug on the body. In essence, what the drug does to the body. These factors impact efficacy and toxicity of drug therapy. For example, a polymorphism may lead to increased drug plasma concentrations, meaning more drug is present in the body. More drug in the body may impact efficacy or toxicity. Pharmacogenomics offers several advantages. The availability of genotyping technologies has made personalized medicine a reality. Pharmacogenomics has the potential to optimize drug therapy in multiple ways, including maximizing drug effectiveness, minimizing drug toxicity, minimizing pharmacokinetic and dynamic variability of drug therapy, and avoiding unnecessary treatment. Pharmacogenomics also has the potential to optimize drug development. Although pharmacogenomics offers the promise of improved care for many patients, there are real challenges with its implementation, including access, feasibility, and cost. Further, the establishment of guidelines for pharmacogenomic-based therapies will depend upon the accumulation of more evidence based upon well-designed studies that continue to emerge. There are concerns, we understand, for both patients and providers, which complicate the implementation of pharmacogenomic-based therapy. Patients often have high expectations, assuming their healthcare providers will explain and interpret pharmacogenomic tests for them. Providers may lack evidence-based resources at the point of care to guide them in clinical decisions. They may be reluctant to order pharmacogenomic tests. There may not be places or labs where providers can order labs to or from, getting the lab tests from. There are health informatics tools such as electronic medical records and computerized provider order entry systems that do not yet have pharmacogenomic information available at the point of care. Both patients and providers have concerns about privacy issues as well. So currently there are healthcare professionals that express the need for more education, and pharmacogenomics curricula have become available and have increased the educational materials in pharmacy schools as well as in medical schools. Experts recommend that training in pharmacogenomics include the genetic basis of disease, the impact of genetic alleles on drug metabolism, drug discovery, drug disposition and targets, and ethical applications, including social and economic implications. And experts also recommend for the availability of open access, comprehensive web-based tutorials and educational materials to be available to all. The field of pharmacogenomics is growing rapidly, with many new discoveries coming to light. It is critical for clinicians to appropriately interpret emerging data on pharmacogenomic tests and to become familiar with resources applicable to their practice.
Now we will review some basic molecular biology, which is the foundation for understanding pharmacogenomics. We will assume an understanding of DNA, RNA, and protein, as well as transcription and translation. These concepts will not be discussed in detail. In this section, we will describe the following concepts: chromosome, DNA, gene, nucleotide, alleles, polymorphism, genotype, phenotype, and haplotype. So, what is a chromosome? It is a long strand of DNA, tightly folded and complexed with proteins into a compact structure. Chromosomes range in size from 50 million to 250 million nucleotides. Humans normally have 46 chromosomes. What is DNA, or deoxyribonucleic acid? It is a molecule composed of two strands of nucleotides associated along their length to form a double helix. What is a gene? The historical definition is a stretch of DNA along the length of a chromosome that codes for a single protein. However, the contemporary understanding of genes recognizes that some do not result in protein synthesis. They may encode various species of functional RNA, or they may regulate the expression of other genes along the length of the chromosome. A nucleotide is a basic building block of DNA. It is composed of a base, a sugar, and a phosphate group. The terms nucleotide and base are often used interchangeably. There are four types of bases: adenine, or A, guanine, or G, thymine, or T, and cytosine, or C. The order of the bases along the length of the DNA comprises the DNA sequence. What are alleles? Alleles are different versions or alternate sequences of a gene at a particular location on a chromosome. Alleles include the wild type, that is the usual sequence, mutations, and polymorphisms of a given gene. Within a gene, variations of an individual nucleotide can be considered alleles. Humans are diploid organisms. That means we normally have two copies of every chromosome. Thus, we have two copies of every gene. One allele is from your biological mother, and one allele is from your biological father. What is a polymorphism? Variations in DNA sequence may be described as polymorphisms or mutations. If a variation is present in 1% or more of the population, it is known as a polymorphism. But if the variation is present in less than 1% of the population, It is known as a mutation. There are different types of polymorphisms. A single nucleotide polymorphism, or SNP, affects only one nucleotide in the DNA sequence. Other types of polymorphisms involve a change in more than one nucleotide. Now we will consider the terms genotype and phenotype. Each person carries 2 alleles of each gene. The set of 2 alleles is his or her genotype. A phenotype is the set of characteristics, for example, clinical presentation, of an individual that result from his or her particular genotype. As an example, variations in the cytochrome P450 genes may result in the contrasting phenotypes of ultra-rapid metabolizers and poor metabolizers of particular drugs. A haplotype is a set of alleles at multiple neighboring positions that coexist on the same chromosome. These alleles may be in separate locations within a single gene or among many genes. Different neighboring alleles, which are located near one another, are physically tethered and usually inherited as a set. That is, their linkage on the chromosome prevents their separation during inheritance. One individual inherits 2 copies of a haplotype. Next, we will cover Pharmacogenomic Nomenclature. This is the pre-test question. The question will be repeated later as a post-test question, and the answer will then be provided. As an example of a SNP is VCOR-C1 1173C to T. Based on the nomenclature of this SNP, what is the gene of interest? Possible answers include A, 1173, B, VCOR-C1, C, thymine, or D, cytosine. There are a variety of naming conventions for gene variations. We will review the following: SNP nomenclature, reference SNP nomenclature, star nomenclature, genotype nomenclature, and haplotype nomenclature. We will start with SNP nomenclature. It is used for naming single nucleotide polymorphisms. This system highlights a change from the normal or wild-type sequence. Our 2 examples are VCOR-C1 1173C to T and ABCB1 3435C to T. The first few letters and numbers, such as VCOR-C1 and ABCB1, are the gene abbreviation. The number after the gene name is the nucleotide position within the sequence of the gene. In these examples, the nucleotide positions are 1173 and 3435. The next letter indicates the original or wild-type nucleotide at that position. The last letter indicates the variant nucleotide, which has replaced the original one. In both of our examples, the original nucleotide, C, was replaced with a T. The reference SNP, or RS naming system, is used by the SNP database. This is the central database for all genetic variation information and is available to the public. The Human Genome Variation Society recommends the naming system of the SNP database as the standard nomenclature for all SNPs. As each new polymorphism is identified, the information is submitted by researchers to the SNP database. The reference data are curated and a computer-generated RS number is created. Star nomenclature is yet another naming system to describe various alleles. Gene variations may lead to altered function of the encoded protein, but the outcome may be different for different genes. In the first example, variations in the CYP2C19 gene result in altered activity of the encoded cytochrome P450 enzyme. For every gene, the STAR1 allele is always the wild type, leading to normal enzymatic activity. In this case, the star 2 allele is a polymorphism which abolishes enzyme activity. In the second example, variations in the CYP2C9 gene result in altered activity of a different cytochrome P450 enzyme. Again, the star 1 allele is the wild type, leading to normal activity. But in this case, the star 2 allele is a polymorphism which simply decreases the amount of enzymatic activity. These examples illustrate that you cannot extrapolate information regarding particular alleles from one gene to another gene. Identical allele names may indicate different functional outcomes depending on the specific gene. Genotype nomenclature describes both alleles, or both haplotypes, carried by an individual. Here, our example is CYP2C19. A person may carry 2 copies of the star 2 allele for the CYP2C19 gene. In this case, their genotype would be described as CYP2C19 star 2 star 2. Alternatively, the person could carry 2 different alleles for this gene. A person's genotype may predict the level of enzyme activity in their cells. A person with a genotype CYP2C19 star 1 star 1 is likely to have normal levels of enzyme activity. However, the genotype CYP2C19 star 1 star 2 or star 1 star 3 is likely to have reduced enzyme activity. And finally, if their genotype is CYP2C19 star 2 star 2, star 2 star 3, or star 3 star 3, there will be no enzyme activity. We next turn to haplotype nomenclature. Recall that a haplotype is a group of multiple linked polymorphisms inherited as a set. Here, our example is VCOR-C1 gene. There are SNPs in at least 10 separate positions throughout the gene that may have functional effects. A unique name may be assigned to each haplotype, simultaneously describing an entire set of linked SNPs. In the case of VCOR-C1, haplotype A defines a particular set of SNPs at 10 different positions along one chromosome, while haplotype B defines a different set of SNPs at the same 10 positions on another chromosome. This is the post-test question. An example of a SNP is VCOR-C1 1173C to T. Based on the nomenclature of this SNP, what is the gene of interest? Possible answers include A, 1173, B, VCOR-C1, C, thymine, or D, cytosine. The correct answer is B, VCOR-C1.
Our next topic is polymorphism types. This section will include 2 self-assessment questions and 2 patient case studies. There are several types of polymorphisms. We have already mentioned the first type, the single nucleotide polymorphism. In addition, there are other polymorphisms which involve more than one nucleotide. These may affect as many as a few or as many as thousands of nucleotides. They include variable number tandem repeats, gene deletions, and copy number variants. Each of these will be described in the next few slides. A SNP is the result of a single base substitution. Several million SNPs have been identified, and novel SNPs continue to be discovered. Some SNPs lie outside the protein coding regions of genes and may alter the expression level of a gene. Others lie within protein coding regions of genes. These may or may not alter protein synthesis. They include synonymous polymorphisms, non-synonymous polymorphisms, and polymorphisms that result in a premature stop codon. This is a pretest question. The question will be repeated later as a post-test question, and the answer will then be provided. A polymorphism has been found in the gene for a drug-metabolizing enzyme. A nucleotide change occurs, yet the encoded amino acid is unchanged. What type of SNP is this? Possible answers include A. Gene deletion, B. Synonymous, C. Non-synonymous, D. Premature stop codon. The first type of polymorphism we'll describe is the synonymous SNP. Our example is the gene ABCB1, which encodes P-glycoprotein. P-glycoprotein is a drug transporter protein found in the cell membrane. The allele we will consider is ABCB1 3435C to T. In this case, the nucleotide at position 3435 is changed from the wild-type C to a T, yet the resultant amino acid, isoleucine, is unchanged from that encoded by the reference sequence. The figure compares the wild-type and variant nucleotide sequences and the corresponding amino acids encoded by each. The underlined red letters are the nucleotides at position 3435 and the amino acids they encode. The codons ATC and ATT both encode the amino acid isoleucine. There are conflicting data about whether this polymorphism affects P-glycoprotein expression or activity. Drugs that are substrates for P-glycoprotein include efavirenz and cyclosporine. The next type of polymorphism is a non-synonymous SNP. Our example is the gene TPMT, which encodes thiopurine methyltransferase. We will consider the TPMT star 3A haplotype, which harbors two SNPs. The G at position 615 is changed to an A, and the A at position 874 is changed to a G. Both of these SNPs change the encoded amino acids. The figure compares the wild type and variant nucleotide sequences and the corresponding amino acids encoded by each. The underlined red letters are the relevant nucleotides within the haplotype and the amino acids they encode. The first nucleotide substitution changes the encoded amino acid from alanine to threonine. The second change that encodes the amino acid is from tyrosine to cysteine. The effect of the variant haplotype is to decrease TPMT enzyme activity. This will impact the pharmacokinetics of azathioprine and 6-mercaptopurine. This slide summarizes the metabolism of azathioprine and 6-mercaptopurine, or 6-MP, drugs which are used in oncology and organ transplantation. Both are prodrugs which are metabolized via three possible pathways. The TPMT star 3A haplotype decreases TPMT enzyme activity. Consequently, more of the prodrug is diverted to the other pathways, one of which converts it to the active metabolite 6-thioguanine nucleotides. By this mechanism, the polymorphism leads to an increase in 6-TGN concentrations, increasing the risk for thiopurine toxicity despite standard doses. The take-home message is that standard doses may not be appropriate for all patients. This is the post-test question. A polymorphism has been found in the gene for a drug-metabolizing enzyme. A nucleotide change occurs, yet the encoded amino acid is unchanged. What type of SNP is this? Possible answers include A. Gene deletion, B. Synonymous, C. Non-synonymous, or D. Premature stop codon. The correct answer is B. Synonymous. Our first patient case will use pharmacogenomic concepts to evaluate the connection between the drug 6-mercaptopurine and the gene TPMT. A 7-year-old Caucasian child is diagnosed with acute lymphoblastic leukemia. The patient has finished remission induction and will begin intensification chemotherapy that will include 6-mercaptopurine, or 6-MP. The patient has no known drug allergies. There are two questions we will consider. Should a genetic screening test be done before starting 6-MP? And what will be the empiric 6-MP starting dose? Effective application of pharmacogenomic principles to clinical practice will require that we implement a systematic approach to understanding polymorphisms. First, we must identify the relevant polymorphism and what it may affect. It may alter a specific protein, such as an enzyme, transporter, or receptor. Alternatively, no functional effect may occur. Second, we must identify which patients are impacted. Genetic variation may exist at both individual and population levels. Third, we should consider whether the polymorphism is relevant to drug therapy. It may affect the pharmacokinetics or pharmacodynamics of a medication. These in turn may impact the dosing, efficacy, or toxicity of the drug, or have no effect at all. Finally, we must consider whether the polymorphism is related to a specific disease condition. It may increase or decrease susceptibility or be useful as a screening or diagnostic tool. Let's apply this approach to our patient case. First, identify the polymorphism and what it may affect. The polymorphism that is relevant in this case is TPMT star 3A, which results in decreased TPMT enzyme activity. Second, identify which patients are likely to be impacted. There is a higher prevalence of this allele within Caucasian populations, where the frequency is 1 to 10%. Third, determine the relevance of the polymorphism to a specific drug. The TPMT star 3A allele may result in increased 6-mercaptopurine concentrations. Higher amounts of the prodrug lead to increased production of the active metabolite. This increases the risk of toxicity, including myelosuppression. Consequently, a 6-MP dose reduction may be called for in patients carrying the TPMT star 3A allele. Fourth, consider the relevance of the polymorphism to a disease. In this case, there is no difference in overall survival in individuals who have the TPMT star 3A polymorphism. Returning to the case of our 7-year-old Caucasian patient, the first question posed was, should a genetic screening test be done before starting 6-MP therapy? Following the systematic analysis provided on the previous slide, the patient was screened for the TPMT polymorphism before starting chemotherapy. The test revealed the patient's genotype was TPMT star 3A star 3A. This indicated that the patient was at increased risk of myelosuppression upon starting chemotherapy. The second question posed in the patient case was, what will be the empiric 6-MP starting dose? To decrease the risk of toxicity, 6-MP starting dose reductions are recommended in the prescribing information. In summary, this case provides an example of a polymorphism impacting drug toxicity. This is a pretest question. The question will be repeated later as a post-test question, and the answer will then be provided. If drug X is predominantly metabolized by the CYP2C19 enzyme, which CYP2C19 genotype may result in the lowest amount of metabolite Y in the blood? Possible answers: A. CYP2C19 star 1 star 1, B. CYP2C19 star 1 star 2, C. CYP2C19 star 1 star 3, D. CYP2C19 star 3 star 3. The next type of SNP we will address is one that results in a premature stop codon within the protein coding region. Our example is CYP2C19, which encodes a cytochrome P450 enzyme. The CYP2C19 star 3 polymorphism is located in exon 4 in the gene. The single nucleotide substitution in this allele replaces the wild-type G with an A. This transforms the codon for tryptophan into a stop codon, leading to premature termination of protein synthesis during translation. The figure compares the wild-type and variant nucleotide sequences and the corresponding amino acids encoded by each. The underlined red letters are the nucleotides at position 636 of exon 4 and the amino acids they encode. The wild-type codon TGG, which encoded tryptophan, is replaced with the codon TAG, which is a stop codon. The functional effect of CYP2C19 star 3 is a loss of enzyme activity. This will impact the pharmacokinetics of proton pump inhibitors such as Omeprazole and lansoprazole. Omeprazole is metabolized by the CYP2C19 enzyme in the liver. Shown are data from a healthy subject study revealing the impact of CYP2C9 genotype on omeprazole pharmacokinetics. Subjects were stratified based on their CYP2C19 genotype indicated in the first column. The second column indicates the resulting CYP2C19 enzyme activity for each genotype. The last column contains the mean omeprazole exposure, or area under the curve. The exposure in subjects lacking enzyme activity was 14-fold higher than that in normal individuals. These results demonstrate that CYP2C19 genotype significantly affects omeprazole exposure, although the study did not indicate whether the difference in pharmacokinetics impacted clinical outcomes. However, clinical outcomes were evaluated in a different study. The authors in this case concluded that CYP2C19 genotypes were predictive of Helicobacter pylori cure rates, as well as peptic ulcer cure rates following dual therapy with omeprazole and amoxicillin. The subjects in this study were patients with H. pylori positive peptic ulcer disease. On the y-axis of this graph is the H. pylori cure rate. On the x-axis are the genotypes of study subjects. The data indicate that H. pylori cure rates are a function of CYP2C19 genotype. Patients with the CYP2C19 *2*2, *3*3, and *2*3 genotypes had a 100% cure rate. Patients with the CYP2C19 *1*1 genotype had a lower cure rate of 30%. Here is our second patient case. A 35-year-old Asian female complains of dyspepsia and epigastric pain. She denies nausea and vomiting as well as blood in her stools. A urea breath test is positive. She is diagnosed with H. pylori peptic ulcer disease. The patient has no other significant past medical history and no known drug allergies. She begins a 10-day course of omeprazole, amoxicillin, and clarithromycin. Questions to consider include the following. What is the primary enzyme responsible for omeprazole metabolism? Does a polymorphism exist for this enzyme? And what is the anticipated effect of a polymorphism on omeprazole pharmacokinetics and H. pylori cure rate? Let's apply our systematic approach to this patient case. First, identify the polymorphism and what it may affect. The CYP2C19 *3 polymorphism, which abolishes CYP2C19 enzyme activity, is relevant to this case. Second, identify which patients are likely to be impacted. The frequency of the CYP2C19 *3 allele is higher in Asian populations than in others. Third, determine the relevance of the polymorphism to a specific drug. The CYP2C19 *3 allele leads to higher omeprazole plasma concentrations compared to the wild-type allele. Fourth, consider the relevance of the polymorphism to a disease. H. pylori cure rates in patients taking omeprazole vary based on CYP2C19 genotype. Returning to the case of our 35-year-old Asian woman with peptic ulcer disease, she purchased a commercially available genotyping kit which revealed that her genotype was CYP2C19 *3*3. Her H. pylori cure rate was anticipated to be 100% based upon her genotype. Upon completion of a 10-day course of the triple drug regimen, the patient's symptoms of dyspepsia and epigastric pain resolve. This case provides an example of a polymorphism which impacts drug pharmacokinetics and pharmacodynamics and is associated with efficacy. The patient was H. pylori negative and considered cured. In summary, this case provides an example of a polymorphism impacting drug efficacy. This is the post-test question. If drug X is predominantly metabolized by the CYP2C19 enzyme, which CYP2C19 genotype would be predicted to result in the lowest amount of metabolite Y in the blood? Possible answers. A. CYP2C19 *1*1. B. CYP2C19 *1*2. C. CYP2C19 *1*3. D. CYP2C19 *3*3. The correct answer is D. CYP2C19 *3*3. Next, we will move beyond the SNP to consider polymorphisms which involve more than one nucleotide. The first type is the variable number tandem repeat. Our example is the UGT1A1 gene, which encodes UDP glucuronyl transferase 1A1. The promoter region of this gene contains a series of 6 tandem repeats of the dinucleotide sequence TA. The UGT1A1 star28 allele contains 1 extra TA dinucleotide, bringing the number of repeats to 7. The figure compares the wild type and variant nucleotide sequences in the promoter region. The underlined red letters are the additional nucleotides in the variant. The altered nucleotide sequence leads to reduced UGT1A1 transcription. Consequently, less UGT1A1 enzyme is produced and there is less glucuronidation activity. This will impact the pharmacokinetics of irinotecan, which is metabolized by UGT1A1. Our last 2 polymorphism types are gene deletions and copy number variants. These both affect the gene CYP2D6, which encodes yet another cytochrome P450 enzyme. Deletions and duplications of the CYP2D6 gene alter the number of copies of the gene, and thus, in turn, affects the activity of the CYP2D6 enzyme. These polymorphisms have as much as a 200-fold effect on drug metabolism. CYP2D6 polymorphisms lead to pharmacokinetic variability among patients, resulting in several phenotypic categories. Patients are classified as ultra-rapid metabolizers, extensive metabolizers, intermediate metabolizers, or poor metabolizers. These data are from a study of dextromethorphan metabolizer status in a small population. Dextromethorphan is a drug that is commonly found in over-the-counter cold and cough formulations and is metabolized by the CYP2D6 enzyme. Among people, there is wide variation in the pharmacokinetics of CYP2D6 metabolized drugs, as indicated by this graph. The number of individuals tested in this study are on the y-axis. On the x-axis is the urinary ratio of dextromethorphan to dextrorphan, which is a measure of dextromethorphan metabolism. The majority of subjects in the general population are extensive metabolizers, or EMs. Drug efficacy may be reduced in ultra-rapid metabolizers because they metabolize dextromethorphan more quickly, resulting in less of the parent drug and more of the inactive metabolite. Poor metabolizers may be more susceptible to drug toxicity because they metabolize the drug more slowly. What is the genetic explanation for these phenotypic differences? A gene deletion affecting CYP2D6 is one explanation for the poor metabolizer phenotype. The CYP2D6 star 5 allele is the result of a deletion which has eliminated the entire CYP2D6 gene. This is a polymorphism involving thousands of nucleotides. The figure shows the CYP2D6 gene in green, positioned on a chromosome among 3 other genes. The variant allele completely lacks the CYP gene. This leads to a loss of CYP2D6 enzyme activity. Affected drugs are those that are metabolized by CYP2D6, including selective serotonin reuptake inhibitors, or SSRIs, tamoxifen, codeine, and beta blockers. A second genetic explanation for variability in metabolizer phenotype is one or more duplications of the CYP2D6 gene resulting in copy number variants. This phenomenon is known as gene amplification. CYP2D6 star 2 times N alleles carry extra copies, where N indicates the number of copies of the gene on a single chromosome. Copy number may vary from 2 to 13. The figure shows the wild-type CYP2D6 gene in green on a chromosome among 3 other genes. Below is the CYP2D6 star 2 by 2 allele, which contains 2 complete copies of the CYP gene on a single chromosome. The functional effect is an increase in the amount of CYP2D6 enzyme, conferring an ultra-rapid metabolizer status. These variants will affect the pharmacokinetics of SSRIs, tamoxifen, codeine, and beta blockers.
Now let's consider some practical issues clinicians face as pharmacogenomics becomes increasingly integrated into practice. As we incorporate pharmacogenomics into practice, a variety of ethical concerns arise. These include privacy issues, also determining who will get the test and whether access to testing is equitable. Although a test may contribute to prevention strategies, there is a potential for misuse of genotyping results to infer a variety of potential phenotypes. Finally, guidelines will be needed to clarify questions about when it is appropriate to order a test and how results should be handled. In 2001, the Equal Employment Opportunity Commission, or the EEOC, filed a lawsuit against the Burlington Northern Santa Fe Railroad Company for secretly testing its employees for genetic predisposition to carpal tunnel syndrome. Although this is a common syndrome, it's a rare genetic condition. Genetic testing for other medical predispositions such as diabetes and alcoholism was also performed. The railroad company employees were not informed of the genetic testing and were threatened with possible termination if they did not comply. So the Equal Employment Opportunity Commission argued that the tests were unlawful under the Americans with Disabilities Act because the tests were not job-related. So finally, the Burlington Northern Santa Fe Railroad Company settled the lawsuit and stopped testing their employees in 2002. In response to concerns about the misuse of genetic information, the U.S. Congress passed the Genetic Information Non-Discrimination Act, or the so-called GINA Act, in 2008. This legislation protects Americans from discrimination regarding health insurance and employment based on genetic information. Legal questions for healthcare professionals include the following. If testing is recommended, are clinicians liable if they do not offer or order the test? If an adverse drug reaction occurs, who is responsible? If you would like more information regarding this topic, the National Human Genome Research Institute and the University of Michigan Center for Public Health and Community Genomics are good resources, and they post a lot of the information on their websites as well. Pharmacogenomics also impacts social issues. Health disparities may occur due to unequal access to testing or the limitations of race-based therapeutics. Genetic tests may lead to employment discrimination, or it may negatively affect insurance coverage. Societal benefits and burdens of testing will need to be assessed as well, and we must carefully consider whether screening is mandatory or voluntary. Finally, there are also economic issues to consider. Implementation of pharmacogenomic tests will require evidence-based rationales demonstrating cost effectiveness. It will also be necessary for payers to agree to cover costs. Studies have shown that the cost of pharmacogenomic tests is unlikely to disrupt the current public health system if the adoption of testing is gradual and incremental. As healthcare professionals, what are our roles in light of recent developments in pharmacogenomics? Well, we can become a source of information by educating ourselves. We do this by identifying published literature and online resources, maintaining up-to-date knowledge, interpreting test results including potential outcomes and adverse reactions. We can share pharmacogenomic information with our colleagues and patients. And finally, we can collaborate with researchers, other clinicians, and educators to translate the best evidence into practice. There are many issues to consider when we counsel patients. These include providing general information about pharmacogenomics tests, assessment of risk in the absence of genetic testing, the cost associated with testing and counseling, the technical accuracy of tests, interpretation of positive, negative, or inconclusive results, the psychological impact of test results, confidentiality issues and risk of potential discrimination, and sharing genetic test results with at-risk relatives. It is unclear how the standard of care will evolve over time. There are still questions in practice that remain. about mandatory testing, restricting the testing, or offering testing so that patients may decide to get the test or not. In addition to genetics, other factors such as epigenomics may become relevant. Epigenomics is the influence of the environment on gene expression. Issues of cost and coverage must be considered, as well as informed consent, patient counseling, confidentiality, and privacy. There are a variety of resources which provide more information about pharmacogenomics. The Centers for Disease Control and Prevention support two initiatives to advance pharmacogenomics. The first is EGAP. EGAP is an initiative launched in 2004 to support a coordinated, systematic process for evaluating genetic tests and other genomic applications that are in transition from research to clinical and public health practice. In addition, the GAP translation programs funded by the CDC Office of Public Health Genomics include these 5 programs on your slide. And all these programs also provide resources either by posting them on their websites or by providing them to their stakeholders. FDA released a final guidance document on pharmacogenomics data submissions in March 2005. In that final guidance document, there were 1,200 drug labels that were reviewed from 1945 to 2005. Of those, 121 labels contained pharmacogenomic information. 69 of these refer to human genomic biomarkers. Of the 69 human genomic biomarkers on reviewed drug labels, 43 pertain to polymorphisms in cytochrome P450 enzymes, most commonly CYP2D6. Currently, the FDA lists over 100 approved drugs with valid genomic biomarkers described their labels. To learn more about definitions, terminology, or the nomenclature of pharmacogenomics, you may visit the websites of the National Human Genome Research Institute or the Genetics Home referenced by the U.S. National Library of Medicine. To learn more about molecular biology and SNPs, you can visit the National Center for Biotechnology Information or see the review article, A Pharmacogenomics Primer, that was published in the Journal of Clinical Pharmacology. There are several websites provided further information for healthcare professionals. The National Institutes of Health G2C2 program, or the Genetics Genomics Competency Center for Education, provides high-quality educational resources for group instruction or self-directed learning in genetics genomics by healthcare educators and practitioners. The CDC's National Office of Public Health Genomics includes material on ethical, legal, social, and practical issues. Technical and practical information is also available at Pharm.GKB, the FDA, and the National Coalition for Healthcare Professional Education in Genetics. In addition, there is information for patients available at the CDC National Office of Public Health Genomics, the University of Michigan Center for Public Health and Community Genomics, and the Genetic Alliance. We would like to acknowledge several of our peer reviewers who had given their time and effort providing comments relating to contents provided on these slides. These reviewers come from the academics or private practice. They include clinicians and researchers. We also would like to acknowledge the PharmGenet authors as well as our consultants and editorial staff for making these slides available. We hope this information is helpful as you translate pharmacogenomic evidence into practice. If you have questions or comments, please email us at pharmacogenomics@ucsd.edu. Our references are all posted on the website as well, pharmacogenomics.ucsd.edu. We at UC San Diego Pharmacogenomics Education Program would like to thank you for taking the time to review this presentation.